federal employees. During the closed session, we'll be discussing the FY17 funding plans that are being proposed by the various teams. These discussions are confidential, so if anyone were to ask you about these conversations, please refer them to me. Also, we are aware of several conflicts that the workgroup members have with specific FOAs, and when we get to those, we will ask those people to hang up and dial in again when the discussion is concluded. Since this is not the first meeting for any of the members of the work group, I'm not going to go through the procedures in any detail, but I just want to remind everyone that this work group does not fall under the Federal Advisory Committee Act. It's not a FACA committee, which applies only to the Institute Advisory Councils. So this work group serves as in an advisory capacity to the Brain IC councils, and it contains representatives from some of the councils as well as at-large members that broaden and complement the expertise of the group. Your role of the work group is to provide scientific input and advice related to funding strategies, initiative planning, and progress in achieving the goals laid out in the Brain 2025 scientific vision. Finally, I want to thank Samantha White for all of her help in preparing for this meeting and her team, and to also note that if anyone is having any technical problems, please email Sam and she and her team will sort things out for you. And with that, I turn it over to Walter and Josh.
Hi, this is Josh Gordon, yeah, I think I met you all at the last MCWG, but I'm the NIMH director, and we're going to start with a brief overview, and then Walter will discuss the budget. So, yep, sure, I can do that. This one's on. Okie dokie. I don't need the notes, it's fine, don't worry about it. Oh, you're trying to get it show up there. Technical difficulties. The state of the brain is good. Strong. There we go. You are seeing it? Okay. So as you all know, the Brain Initiative is guided by the Brain 2025 scientific vision. Its goal is to see the circuits of the brain in action in order to understand how the brain guides behavior, and also to develop the tools to monitor and manipulate circuits both in animal models and in the human brain. Those tools, of course, include developing a census of the cells of the brain and maps of the brain. The first 5 years of the program, which were a little bit more than midway through, is to emphasize, we are emphasizing technology development. And the second 5 years, we're to emphasize discovery-driven science using that technology development. And you'll probably see that this time around and perhaps even more in the future as we develop funding requests that reflect that emphasis change. This is the timeline of the brain. You're mostly all familiar with this. It was announced in April 2013. The first awards were given in September of the following year, and we're now well into our third or fourth year of giving out awards. I can't quite do the math in my head. There are several other aspects besides just the Brain Awards. We've had a neuroethics division that was established in August 2015 that has been very active of late. In fact, we just had a meeting last week at Duke where we discussed the neuroethics of ex vivo brain tissue. And more such meetings are planned, as well as guidelines to help our investigators know when to raise questions about neuroethics for the divisions and the directors to consider. We also have we also mark the beginning of a Brain Initiative Alliance website that's powered by our partners, such as the Kavli Foundation, and of course we noted this year, late last year, the signing into law of the 21st Century Cures Act, which ensures funding going forward through 2025. These are the last several years worth of appropriations. You'll notice The big jump in fiscal year 2017, we have Congress to thank for that, in addition to the $10 million that were provided for by Cures and the $140 million that were embedded in the appropriations to each of the institutes last year. They gave us an extra $100 million this year, so we should be able to fund a lot of wonderful science based upon that. This is a chart of the yearly funding that's set aside in the Cures budget. It does not reflect the amount that we have in our base currently, which, as I just showed you, is about $240 million, that upper right corner. Let's see if I can show it. We presume that this will be in our base going forward, although obviously it's up to Congress to decide whether to continue that. But on top of that base, Cures provides accelerating dollars through 2026. You notice in fiscal year 2023, there's a spike in funding, and Walter will go into that a little bit later in terms of our plans to smooth that out, that we've worked out in consultation with the what was it, the Office of Directors Council, whatever that's called, the Advisory Council to the Director. Funding opportunities issued for 2017 include cover most of the areas of the BRAIN initiative. 4 of them build on our cell phenotyping efforts to really try to ramp up the development of a cell map. 2 of them focus on training. 3 are aimed at really, for the first time, building data informatics infrastructure to store the data and determine the data elements that will go into those data storages. 2 support small businesses via the BRAIN Awards for Small Business Innovation. 1 addresses neuroethics questions. This is also new for us this year. And 4 create a new brain circuits program. Many of these opportunities have been reissued from 2016, and these include supporting imaging and non-invasive neuromodulation in humans, as well as tool and technology development across all scales and models. And I want to emphasize that these funding announcements span all 7 areas that were identified in the BRAIN 2025 initiative, and also that we are beginning to assess the BRAIN initiative. Our first attempts were to make sure to identify gaps within these 7 areas that need more work, and you'll see some of the product of that in terms of the development of the funding requests that we're presenting to you today. And we're planning to revisit BRAIN 2025 a little bit more extensively to get input on what fundamental questions about the BRAIN initiative could or should focus on, given the advances that we've made over the last several years. And that process will probably be taking place over the next several years. I forget when you're supposed to come in, Walter, but I might as well just say that there's been lots of wonderful publications that arise from the BRAIN, and we're looking forward to continue those exciting advances. Are you ready to go then? Should I hand it over? I'll hand it over now to Walter Koroshatz, Director of the NINDS.
Great, thanks, Josh. So as mentioned, we are looking carefully now at what the BRAIN initiative has done and trying to plan for the second stage, and with consultation with Dr. Collins' office, plan to try and develop a process similar to the 2025 report to give us advice on the second half of the BRAIN initiative. And but we are really excited about what's happening. The science that's being published is really quite outstanding, and I think that a number of the aspirational goals that people had in the beginning but seemed, quite far in the future now seem to be within grasp. So we're quite, we really feel quite good about what's happened so far. Over 190 publications have emerged from the BRAIN. We have talked in the past about the fact that the BRAIN initiative, because it is getting at some of these unique properties that make us human, bring some interesting neuroethics questions to the forefront. And as Josh mentioned, we have the neuroethics division of the multi-council working group. There have been a number of off-site meetings about different topics, and the one we had most recently at Duke was on the use of ex vivo human brain tissue in research. And our plan is it's a complicated diagram, but our plan is to continuously look at the issues that come up in terms of neuroethics, bring these through the multi-council working group and the IC directors to develop policies when necessary. And with that, I'm just going to, Susan did introduce herself to begin with. She is now the new MCWG Executive Secretary. She talked about what she does at NIDR, but I think the most important thing she didn't mention is she's from Brooklyn, and so that ensures success. Okay, so I think what we'd like to do now is, because we have so much on the agenda, is to move very quickly into the concepts that we want to bring to your attention, and these will require votes. So do we have a quorum is the question. Yeah. We do have a quorum, great. Okay, should we ask, do you want to announce who's on the phone? Or have them announce? Do we know who they are?
No, we know.
Okay. Great.
So of the multi-council working group members, we have Drs. Brad Hyman, Bruce Rosen, Christoph Schreiner, Eve Martyr, Jim Eberwein, Larry Abbott, Mark Schnitzer, Rafa Justa, Richard Huganier, and Story Landis on the phone, as well as Mr. Hank Greeley. Great. If there is anyone else, please chime in or send me a message because you are not showing up.
Okay. Okay, so for concepts, hold on, we'll get the next slide set up. I guess while the slides are coming up, I just say in terms of the budget spikes, for those who are NIH aficionados, what we're doing is getting permission to do full funding of grants so we can actually fund the total time of a grant, 3 or 4 years, whatever it is, and mix that in with our usual funding, which is we fund year at a time, to smooth out and strategize with regard to how we deal with the ups and downs of the Cures funding. So we feel pretty secure that we have a good handle on being able to move into that second stage. So with that, I want to turn over to Team A, Oliver, to talk about a new concept to kind of bring in the non-neuronal cells into the brain initiative. Great.
Hello, everyone. So I'm a brand new program officer with the Brain Initiative, so I'd like to introduce myself briefly. My name is Olivier Burton, and before moving to the dark side, I was a PI at UPenn and Mount Sinai for about a decade. And here I will be I'm participating to the Team A and Team D efforts. And so today I'm happy to present this concept for a new funding opportunity entitled Tools to Enhance Studies of Non-Neuronal Cells. So, which we think would fill an important gap. So why is there a need for such a funding opportunity? Well, the aim would be to stimulate grant application that develop cutting-edge technologies that would be tailored to study non-neuronal cells. So why is there a need for this opportunity? So non-neuronal cells represent the majority of the cells in the nervous system, but they are largely understudied by neuroscientists, and this is partly because there is currently a lack of adapted tools for conducting these studies. Interaction of non-neuronal cells with neurons impact many key functions, such as the formation and control of the blood-brain barrier, the control of blood flow and oxygenation, which are the basis for both signal, and also influence synaptic plasticity, for example. Tools that are developed for neurons, such as sensors and viral vector, often are not directly applicable to the neuronal cells. And so although previous brain FOAs have never formally excluded neuronal cell types from their focus, none of the existing tool development and census activities have so far covered non-neuronal cells. So there seems to be a great need for an opportunity that will be tailored to study those type of cells. So this is not a new idea. Actually, the importance of including non-neuronal cells in brain census activity has been underlined from the outset by Brain 2025, as well as and by the NCWG, as well as by other scientists. And this is, for example, an excerpt from a comment article that was published in Nature in 2013 by our intramural colleague Douglas Fields, who stressed and urged the Brain initiative to not forget glia. So there's a number of aspects that need to be defined regarding the scope of the FOA. So one first aspect is which non-neuronal cells are we talking about? So we propose the FOA to cover all the glial cells, which comprise at least 6 subtypes, including astrocytes, microglia, oligodendrocytes, ependymocytes, radial glia, and NG2 cells, and also to cover vascular cells, which at least comprise 3 different types, including endothelial cells, smooth muscles, and pericytes. And we also, there will be also a strong interest in covering interaction between these different cell types. Which type of tools are we interested in here? We will be, so here a few examples, new genetic and non-genetic tools for delivering genes, proteins, or chemicals to cells of interest, new tools for identifying and classifying non-neuronal cells from a molecular profiling standpoint and morphology standpoint, new tools for manipulating and monitoring their specialized function and investigate their contribution to normal and abnormal neural network function, and intersectional approaches that target specific cell types during defined developmental periods or specific microdomains at the neuroglial vascular interface, such as the end feet of astrocytes, for example. So to conclude, I wanted to reference 2 recent publications that illustrate some of the barriers in this field and also some of the opportunities for new tools. So the first article is an article that was published in 2015 in Neuron by the group of Jaime Gertzendler, and this paper investigated which vascular cells, either smooth muscle or pericytes, mediate neuronal activity induced increase in cerebral blood flow. And this is a question that remains controversial, in part because the tools available to address these questions have a lot of limitations. And so, for instance, viral vectors often that are used to study neurons do not transduce vascular cells effectively. And also the mouse drivers, genetic drivers that are used are actually not specific for different vascular cell types. So, for example, here the investigator used the NG2 Cree driver line that drives transgene expression both in smooth muscle cells and pericytes. And so here, in order to decide which cell to collect calcium signals from, they had to make educated guess about which cell they're collecting signals from based on morphology and localization of the cells. So although those tools have been useful, there's a critical need here for better entry points to study those cell types. The second paper I wanted to mention here is a paper that was published in March in Nature Neuroscience, and that identified new subtypes of astrocytes in the mouse and human brains. And so here they use a combination of genetic driver, which is the classical driver for astrocytes, and also membrane proteins to do fast sorting of different cells, and based on that, they identify 5 different subtypes of astrocytes that they call from A, B, C, and D. And what they show here is that those cells are differentially expressed across the brain, and they have different functions. For instance, they influence synaptic plasticity differentially. And so, here, based on the profile they have obtained, we have a nice illustration of the diversity that exists in astrocytes, but also a nice illustration of new opportunities for using this profiling data to develop new tools to characterize the function of these different astrocyte subtypes. So I'll conclude here, stop here, and take questions if there are any.
I just wanted to ask if anybody has questions for Dr. Berton, if you could just identify yourself when you're asking your question. And you might want to unmute yourself as well.
This is Dory. Am I unmuted?
Sound good, yeah.
So when the BRAIN initiative was first developed, the notion was that it was pretty important to focus it and that there would be lots of parts of neuroscience that wouldn't be included. And I can remember a discussion with Society for Neuroscience members, where's the stem cells, where's the development, where are the disease models? And the NIH pretty much stuck to the notion of a focus on circuits. And it strikes me that while there are many good reasons to want to know a lot more about glial cells, this would move the BRAIN initiative into areas that maybe weren't initially focused on, sort of brain as tissue, and I'm wondering the extent to which that may or may not diffuse the focus that was so tightly placed on circuits, the participants in the circuits, and the function of the circuits and behavior.
Yeah, thanks for that comment, Story. This is Andrea Beckel-Michner, and we're really thinking about this announcement in the context of the cell census activity, where we are looking at all of the cell types, including some of these so-called support cells that influence the behavior of circuits. So we can absolutely be more specific in the announcement if you all think that's useful.
I'm just a little worried that this could get out of control. All those thwarted, frustrated glial biologists feeling that now is their opportunity to learn anything they ever wanted to know about glial cells. Maybe I'm being unnecessarily pessimistic and cynical, but I think it would be great for this to actually be pretty focused and not too expansive. But this may be
Hello,
a
can you hear me?
opinion.
Hi, this is Rick Hugener. Can you hear me?
Yes.
Rick, we got you. Yep.
Okay, good. So Story, you and I rarely disagree, but I mean, I think the key here is how non-neuronal cells affect circuit function. So, you know, there is certainly now we know there's a lot of neuromodulation going on. There's all sorts of interactions with glia, with synapses, and so I think if we focus RFA to that direction, and as you say, sort of try to prevent it from going out of control, focus it on how glial interact with circuits, because I think they're integrally involved in circuits in many, many ways. So
that would be fine. That would be fine with me. But I just worry a little bit about how it could be hard to define the limits of this interest in a way that doesn't end up having it eat the whole BRAIN project.
Okay, I think that, so it sounds pretty clear that if we really focus it down on the glial role in brain circuits, which would include trying to understand the different types of glia so you could actually then get in and manipulate them, actually uncover what they're doing, in terms of the circuitry, that would be fine. And so Story, they pay me to worry now, so I can do the worrying.
Walter, that sounds great. I'm happy now.
Thank you.
Should we just take a vote then? Okay, so okay, so is there anybody who would object to our going forward with this concept? Okay, we'll take that as clearance.
Focused concept.
Focused concept. Thank you.
And just while there's a lull here, I'd also say that Blueprint is actually thinking of projects that would focus more on the general biology of the neuroimmune and the vascular system in the brain. So that will be clearly separate type from what the Brain Initiative is doing. Complementary.
Okay, good afternoon, everybody. I'm Michelle Freund. This is a second concept clearance from Team A, and we're looking for your input on this concept that will solicit applications to develop tools to facilitate high-throughput microconnectivity analysis. Although this was not excluded from the previous tools FOA, we did not receive enough sufficient number of applications that directly address this. So we're hoping we'll target that audience and get some in that are specifically focused on this area. This is in response to the 2025 report focusing on developing maps at multiple scales, generating circuit diagrams that vary in resolution from synapses to the whole brain. As you may remember from some of the applications in the previous tools FOA, we have awarded a number of applications that are looking at long-range connections and sort of medium-range connections, but not really focused so much on the EM level, and that's what this is supposed to facilitate. It remains unclear how variable microconnectomes will be among individuals within a species or even over time in a single individual. This variability would not be an insurmountable problem if it were feasible to routinely map microconnectomes on the same individual that have been analyzed physiologically or compare normal and pathological tissues in a substantial number of individuals. But this throughput is just too expensive with the current set of tools that we have. So the deliverables we would be looking for are improvements in the resolution and accuracy of imaging to enable complete and accurate mapping of neural circuit structure in human and animal brains, integration of structural data across scales from whole brain to ultrastructural and with other modalities, including physiology, molecular biology, and biochemistry. And of course, discovery of anatomical differences between healthy and diseased brains would be a goal. The short-term goals of this would be to develop a high-quality toolbox of methods to efficiently map and annotate projections in experimental animals, including non-human primates as well as in blocks of human tissue. An important area that I think we really want to focus on is to reduce the time needed to segment volume EM data sets by 100 to 1,000-fold, and to reconstruct microconnectomes of individual animals that have been studied physiologically and behaviorally. And finally, to develop new techniques for using EM and or other super-resolution light microscopy to integrate molecular signatures of cells and synapses with their nanoscale connectivity. This funding initiative will focus on the development, optimization, and scaling of the following: techniques for tissue sample preparation and image acquisition, methods for incorporating multimodal information into microconnectivity assays, including information on cell types, gene expression, and synaptic phenotypes, tools for enhancing and scaling the automated imaging processing, connectivity analysis, and data interpretation, including algorithms, information extraction routines, and user interfaces. We'd also like to encourage the development of data sets that can be used to serve as ground truth for algorithm development and testing, and proof of principle integration of structural microconnectivity with functional data at cellular, synaptic, or neural system levels for understanding whole circuits from micro to macro scales. and that is it. So this is a concept that has been discussed previously at MCWG meetings, and we've, I think, listened to your advice, and that's why we're coming to you with this today. So hopefully there won't be any problem.
Do people have questions or advice?
So this is Mark Schnitzer. I have a question about this one. So one reaction that I have to this is that the potential FOA may address a very small body of researchers. I mean, you mentioned a little bit of super-resolution light microscopy at the end. It seems, I mean, yes, that's in there, but it does seem that there's a strong notion from the get-go that electron microscopy is going to be a major way forward. That may well be the case, but I'm just wondering if it's if the FOA is optimally tuned to have it so narrowly focused on EM.
I think that our aim is to have it not so narrowly focused, to include options for other approaches, and we'll wait to see what the community comes up with. We took EM out of the actual title of the FOA.
I think one thing, this is Rick Hugener again, is that this may bring in, I presume you're targeting computational people as well, right? So
included, of course.
Yeah, so there might be a lot more people out there who may apply in that regard, and not so much in the EM aspects, but in the analysis.
That's right. If we had that data set available that we could have the community use, then we would really expect to get some good computational applications.
Well, one question about that would be, which may depend very much on how you write it, would be, the distinctions that people tend to make between connectomics and projectomics. So, at least to this point, some of the optical approaches have been better suited for, mapping projections rather than real, point-to-point synaptic context. So, so one question is, whether the FOA might be written such that it might spur the community to actually help the optical approaches take that next leap, so where they're really competitive, with connectomic studies. I mean, I think what you get may depend very heavily on how you write the challenge.
Absolutely.
Mark, could you be more specific about what the other techniques you would want to have included in this? Well, I mean, I think as as we've all seen, there have been lower resolution light microscopy techniques, many of them variations on the theme of light sheet, but there are others. And by and large, they've not had the optical resolution to resolve, true synaptic context where you can unambiguously identify a presynaptic and a postsynaptic partner. They tend to be better for mapping projections in an intact, three-dimensional brain. So they don't, provide the connectomics at the moment in the way that that the electron microscopy does. Nevertheless, there is reason to think that improvements on what we have now in that lower resolution optical approach might genuinely allow you to get to two synaptic point-to-point context, provided that there was suitable development of paired markers, on the presynapse and on the postsynapse. And, there are many potential benefits of doing it optically if one could, namely throughput, and namely also the fact that you could then leverage the many other optical labels that we have to label genetically defined cell types, to label macromolecules, and so forth. So if we could tackle that connectomic problem in the optical domain, I think it would bring a lot of benefits, and it might be worthwhile broadening the scope here slightly to really encourage innovation in that way.
Hi, this is Eve. Can you hear me? Hello?
Yes.
Yeah.
Yes.
So I really hate to do this, but I agree with Mark. And I think that there are two things I'd like to say at this point, which is in another year or so, there should be already plenty of volumes of EM data that could be used for whatever algorithm testing, et cetera. I mean, the data sets at the EM level are starting to become more and more prevalent, whether it's in flies or whether it's in piece of mouse, mouse brain, et cetera. So I think there's tremendous value in EM connectomics in times and places, but I think from my point of view, looking from the outside, really high quality, high resolution, light level stuff should be able to frame questions so that people could go in a much more targeted fashion to do the very detailed EM connectomics when there's really a point to it, rather than thinking about doing the whole mouse brain at the EM level brute force. So I really think that reframing it a little bit to make sure that people try and get the highest quality, light level, I don't know what you want to call it. I call it still connectomics first, and then to use some of the EM data sets that we already will have to work on the algorithms that would make all the segmentation and all the reconstruction that much faster and that much better.
So if
I think some of Eve's work actually makes this point very clearly. I mean, some of our initial discussions with Brain 2025 emphasized, not only the commonality between different brains of the same species, but also the individuality. And so having the throughput to be able to do multiple brains and not just one mouse is going to be really key if we're going to get at issues of individuality.
Absolutely. And I think that there's tremendous amount that could be done in small pieces of the fly brain, for example, where there's already a tremendous start in connectomics. But, yes, it would be really nice to see 10 examples of a certain region, and therefore the throughput is really, really important. But on the other hand, the whole mouse, you really are going to want to see where cells go and who they talk to, and about how many times they make synapses with what kinds of cells. So I think it all needs to be done, but I think it needs to be done quite strategically.
So I think we appreciate the conversation here. In the interest of time, sorry, this is Josh. In the interest of time, I just want to make sure there are no comments on other aspects of the proposal.
So this is Story, and I would just like to channel David Tank, who for multiple MCWG meetings has argued strongly for an initiative like this. So I would like to see perhaps it expanded, but certainly not throw out the EM with the bathwater.
Well, like Andrea mentioned, the new title is Tools to Facilitate High Throughput Microconnectivity Analysis, and I think that accomplishes both the goals that the people have been arguing for, and we can make sure when the wording goes out that we don't prejudice against EM. So with that,
Great.
then I'd like to ask, yeah, sorry, go ahead.
No, just thanks.
Okay, good. So with that, I'd like to ask if we can, Susan, you want to ask for a vote?
Yeah, are there anyone who objects to the concept being put forward with some of the recommendations for modification that we've been discussing? Okay, wonderful, it's cleared.
Thank you very much, Mark, and I didn't catch the name of the other person who was commenting. Eve, thank you very much. And Story for Tank? Okay. We're queuing up the next one here. Maybe we should queue up the...
Can I ask a question before you go forward? Is there anything specifically in that concept that talks about finding ways of visualizing electrical synapses?
It's not written yet. We can certainly include that as a bullet point.
Because there's a big, big, big need to have new tools for visualization of gap junctions and electrical synapses. It's actually a great problem, and slightly different tools are going to need to be developed for that.
Thank you.
Thanks. So on to the next concept clearance proposal we have for you today. My name is Nick Langhals. I'm Program Director at NINDS and co-lead of the Team B that covers the invasive technologies. And for those that have had the chance to review the concept clearance, the goal here is to develop a new RFA in the area to try and understand the biophysics of neuromodulation. And really what we're trying to get at here is that there are many different tools and technologies that have been developed over the years and are currently still even in the early stages developed, designed to stimulate or modulate the nervous system and interact with the brain tissue. And these could be some of the more frequently used technologies, including TMS or deep brain stimulation, or it could be even more in your intracortical electrical stimulation technologies, ultrasound, or using light-based interfaces. And really the gap that we're trying to address is that we don't necessarily have a good fundamental understanding of how these different fields interact with the underlying neural tissue. And so what we really want to be able to study and help elucidate is really understand those mechanisms of activation, what goes on when these fields are applied at that cellular level of interacting with the tissue. And these could be including things looking at the activation thresholds of individual cells, changes in subthreshold activities that are occurring, changes in morphology that are occurring either acutely or chronically with multiple or prolonged stimulation, changes in metabolic activity of those cells, as well as the cell-cell interactions. And our goal here is in terms of understanding the effects of the neuromodulation, tying it back to the earlier one, we're really still open to including the idea of neuronal and non-neuronal cells, as they're likely going to be some sort of interplay between them when applying these neuromodulation fields and trying to interact with the tissue. Obviously, as many of us know, that when dealing with animal preps, the overall behavioral state, whether they're awake, anesthetized, or paralyzed, could have an effect on those sorts of activity within the brain. So we're open to considering those different conditions, but really we want to make sure that the goal is focusing on that micro scale where we can sufficiently characterize, model, and validate these fields that are being produced from those technologies. We're envisioning this being at that micro scale of a few cells or a small simple circuit of cells, starting for the kind of resolution level that we're expecting. Looking at these small levels and the micro circuits, we're expecting that some of the experiments may be in cell culture, some may be in tissue slice prep, some will be in vivo as well, and hopefully the goal would be to be able to expand and understand that model across those spatial scales when applying these various different fields. How does this fit into the overall Brain 2025 vision? Per the section noted on the screen there, one of the goals is to develop new and improved perturbation technologies that are suitable for controlling cells within the nervous system, that include tools for stimulation, inhibition, or modulation. It could include any of the various different modalities, including optical, chemical, electromagnetic, biochemical, or others. Overall, one of the core principles identified by the MCWG is that there really should be a validation of all technologies. And obviously, when pushing the technological envelope, we need then these new tools and understandings of how those work to then be able to advance them to the next level going forward. Within the overall impact, we really want to then be able to develop these models across the different modalities, across the different scales, and to accomplish that, the plan is to then have the investigators funded under this participate together in a consortium where they're sharing the data and working together to the extent possible to share common data across the different modalities, as well as to then be able to hopefully develop better understandings of the mechanisms of the neuromodulation, and then provide that important test information to then guide and further develop next generation technologies. Within the Brain Initiative right now, several of the RFAs that we have are highlighted on the board here, including an R21 for new concepts for recording and modulation. We have new tools and optimization for modulating technologies covered under some of our Brain Team B U01s. We have various different tools, development for new tools, mechanisms, and dose response under our non-invasive neuromodulation RFAs, and then we're also developing next generation invasive technologies for the human CNS. So with all of these different tools and technologies, they could all take advantage and be able to be accelerated by the advancement of having these better understandings at the cellular level. Some of the examples of in-scope activities may include characterizing the cellular biophysics in the appropriate systems, potentially looking at the safety limits of some of these applied neuromodulatory fields, developing the field modeling for the different stimulation intensities, getting and developing both the spatial and the temporal models whenever possible across the scale to understand what's going on in the tissue, as well as kind of understanding the dose response curve of that electrical stimulation applied. And so as one can imagine, to accomplish a lot of these steps, it likely would involve very multidisciplinary and collaborative teams that include both cellular and molecular physiologists, biologists, neuroscientists, as well as your technology developers to then integrate with those teams, and then the computational side as it relates to developing and integrating those models across the teams. So try to keep it short and sweet, but we're happy to open it up to any questions that anyone has about this concept.
This is Eve Martyr. I find this concept very confusing. And the source of my confusion is twofold. Firstly, the term neuromodulation means totally different things to different communities. And you're using it in a way that the cellular community won't understand. So they mean one whole set of things about when you say neuromodulation, and the high-level systems community means totally different things. So I think you have to, at some point, confront that, or maybe you don't. But I know when I hear neuromodulation by the systems community, it doesn't resonate to anything I know using that word. But that's a minor point. The major point is when you talk about doing the studies in small systems or tissue cultures or even slice, it's not totally clear to me how well the data you get are going to generalize past some of the things that you would want to know if you're going to try and stimulate a human brain versus a mouse brain versus a leech. Some of those things are going to be very tissue idiosyncratic. And so I guess I'm less convinced here that everything that you think you want to do is going to give you general or generalizable answers. I could be wrong. I'm often wrong. And certainly the future is the future, and I'd be happy to be wrong. But I think there's part of this which is a little fuzzy.
Thanks, Eve. And I guess one of the problems or the difficulties. Still green. No? Okay. Sorry. So thanks. So one of the issues, and I agree, we're not necessarily sure how well a lot of these concepts and the details would scale across the various different levels and whether or not they would generalize. But I think, to me, that kind of highlights the gap in understanding that we have of how these different fields are being applied to the tissues, and to understand them, just building up our base knowledge of that interaction could then in the future help to be able to determine whether or not they generalize and what are the appropriate models to understand and validate this. And Holly also wanted to chime in as well.
You're right. This is Holly Lismy. I just wanted to add to what Nick was saying, that we're currently using these neurostimulation tools to do neuroscience studies and to treat disorders, and we're doing that in absence of knowledge of the mechanisms at the biophysical level. So even if we were just doing it in the species that were already using these tools, then it would generalize to the experiments that we do in those specific species and help us understand what we're learning from the experiments we're currently funding today.
This is Larry Abbott. I wanted to just mention, I thought the language, at least in the modeling, seemed like you wanted to stress the biophysics end and not the system circuits end. But, that's the barrier that I think we should be trying to knock down, a set of people that look at modulation in cells and another set of people who kind of ignore it. So I would hope you'd stretch that out a little bit.
Yeah, this is Mark Schnitzer. I was going to make a similar comment, namely that with all the emphasis on the Brain Initiative on circuitry, and with the fact that we now have cell-type specific ways of stimulating circuit activity, this is, very different from the kind of more macroscopic application of field stimulation that is commonly referred to as neuromodulation. And without making any bets about what the long-term, future of this is, I think one has to agree with Larry that trying to understand the differences between cell type specific modalities of stimulation and general application of field, I think is going to be critical. So when I hear biophysics of neuromodulation, I think just about the application of field without regard to the specificity that we have at our disposal nowadays.
Well, and I guess I agree, and we weren't necessarily trying to exclude that. I think our thought is that we really do need both that understanding of the field modeling as well as the understanding at kind of that microcircuit level and those interactions. And as you mentioned, I mean, we have all these new tools that allow us to then be able to better record those sorts of small level interactions and activity that could then be taken advantage of potential applicants in understanding these.
Yes, and we also wanted to not overlap with our other existing funding opportunity announcement, which does link neuromodulation to mechanisms of action at the circuit level, but that's really macro circuit. I think this concept could be expanded to include the microcircuit level to address the concerns you're raising.
Rick Hugener, I think I see that your mic is on. Did you have a comment?
No, I was muted.
Oh, sorry. Holly used a word that's different from neuromodulation, which is neurostimulation, and although I realize that you might think that things also work by neuroinhibition, is neurostimulation generic enough but better than neuromodulation if we're concerned about confusion?
It certainly would be less confusing for me, or at least to make sure that you somehow or other acknowledge the fact that if you go down to a slice preparation and the people who study the biophysics of ion channels and slices, they're going to understand something completely different when you say neuromodulation of the action of circuits in a slice than the people who you're really trying to target. Now, and I also think, I mean, I feel very, very strongly that 3/4 or large numbers of people who use optogenetic stimulation right now have no idea actually what they're doing in terms of, so defining better what the methods are already in use, what they're actually doing is very, very important. But again, I come back to the notion that everybody has to do the right controls and the right measurements in the experiment that they're doing. And so I'm cautious about how generalizable. Some things will obviously be very important and very generalizable, but lots of other things may be very, very specific and necessary controls to the local community that's using those methods. So I think it's just going to be tricky when these proposals come in. I can imagine lots of different kinds of things coming in where they all think they're responsive to this, but they're actually responsive to something that they're reading with a very different optic.
So do you think then that we should go forward with this concept? Do you want it to be presented again in August with a little bit more of a specificity?
People to say, yeah.
I think that's a good idea to rethink it and maybe reword it and come back in August, because you could still do it and have it funded in 2018.
Okay, so is there anyone that objects to postponing this or rehearing this again at the August meeting? Okay, then I think that's where we are, and we'll go to the last one by Holly Listenby.
Okay, good afternoon. Can everyone hear me okay? My name is Holly Listenby. I'm the Director of Translational Research at the National Institute of Mental Health, and I'm presenting the concept on brain behavior quantification on behalf of BRAIN's teams B, E, and the Office of Behavioral and Social Sciences Research. A major function of the brain is to generate behavior. These behaviors include our thoughts, our feelings, our cognitions that form our inner mental experience, that make us ourselves, that comprise the human experience, as well as behaviors that are externally facing, that allow us to navigate the world, allow us to communicate with each other, give talks like this, have relationships, and lead rewarding lives. And in fact, it's a disruption in the brain's ability to generate these higher cognitive aspects of behavior that result in the leading causes of disability worldwide in terms of neurological and psychiatric disorders. So it's not surprising then that behavior plays a central role in the goals of the BRAIN Initiative, and specifically in the BRAIN 2025 report. These are some quotes from the report which illustrate that the BRAIN Initiative was really launched at its basis to discover how the brain generates cognitions and behaviors at the speed of thought. It posits that a leading purpose of the brain is actually to generate these behaviors. And you can, if you search the report, you'll see that throughout the report this is emphasized, and indeed it's said that the most important outcome of the BRAIN Initiative will be a comprehensive understanding, mechanistic understanding of mental function and how it arises from the brain. So understanding the brain at the speed of thought, which is certainly a wonderful rallying call for us working in this field, is a tremendous challenge, especially when you consider speed. This means that we need to match the temporal resolution of our brain recordings with the temporal resolution of our behavioral recordings, and indeed to do them simultaneously so that we can establish causal links. That being said, it's somewhat surprising that only one of the projects that the BRAIN Initiative is currently funding achieves this goal by studying behavior and measuring behavior simultaneous with brain recordings. This proposal seeks to fill that gap. It will be the first BRAIN Initiative funding opportunity announcement that includes direct recording of behavior in real time simultaneous with brain recordings. The goal here is to link quantitative and temporally rich measurements of behavior that are objectively acquired during brain recording at the neural circuit activity level. This goal of the project is to align fields that are convergent and yet infrequently collaborate, advances in cognitive and behavioral neuroscience along with the cutting-edge neuroengineering, neurorecording, and modulation community. Part of the project is to connect these research communities to develop multidisciplinary teams that would be able to work together to achieve these goals, to radically transform how these research paradigms are done. This will leverage new advances in mobile technologies that are already available and will stimulate the development of new ones that are able to link the technologies for behavior quantification with the new technologies we have for brain quantification, as well as with advances in computational approaches to make sense of the data. So the goals of the initiative are to develop and link quantitatively temporally dense behavioral measures to neural circuit activity. To establish these linkages and to link the technologies, basically enable new tools that are able to bring together these multiple sources of data and literally put behavior in the loop. Many projects have a closed loop approach, but really they're closing the loop between one part of the brain and another. Here we're seeking to close the loop between brain and behavior. What would that look like? Well, here is a schematic. That technology already exists for direct recording of neural activity from the brain. This recording can be linked to simultaneously measured and quantified behavior using mobile technologies, which probably you have on your wrist or you're carrying in your pocket right now. And these can be linked using wireless links that allow the real-time capture of behaviors while the brain is being recorded. This can be 24/7, it can be in realistic environments, it can be at home, and to be able to link these recordings on the same time scale. Since this allows us to sample throughout the day, we can understand changes in state, we can understand changes in alertness, factor in chronobiology, time of day, and because it's linked to electrodes that are directly recording from the brain, we can also stimulate the brain circuits to establish causal relationships between the brain circuits and the behaviors arising from them. So why do this now? We have advances in technologies both for recording the brain activity as well as for objectively measuring a broad range of behavioral outputs. And we have advances in computational approaches for hosting this data, for analyzing, and for mining this data to gain mechanistic insights. We are currently funding studies that give us high temporally dense measures from the brain. However, as you've seen, there's a paucity of studies that also measure behavior at the same time scale. So it leverages investments we've already made in the brain instrumentation and brings to it the cheaper and currently available behavior quantification measures. And it's very aligned with some council work group meetings that we've had at the National Institute of Mental Health and at the Office of Behavioral and Social Sciences Research Council work group that have emphasized the importance of incorporating mobile technologies to enhance human health. So why should this be part of BRAIN? It can be added on to projects that are already part of BRAIN. We're currently funding human invasive technologies that give us high-quality direct neural recordings and allow us to intervene in these circuits by stimulating to change behavior and establish causality. Of the human projects that we're funding, the majority of them have behavioral measures. However, only one of these have behavioral measures that are acquired simultaneous with the brain recording at the same temporal resolution. What do we mean by behavior? Well, there's a broad range of things that could be responsive. Of course, we want to measure behaviors that are expected to arise from the brain circuits where the recording electrodes are located. This could include movement, motor control, eye control, sleep changes in consciousness, language processing. It could include affective expression, cognition, many of the things that are important for human health and are affected by disease. We have already existing tools that allow us to quantify a broad range of behaviors, from motion sensing to sensing social interactions that are quantified conveniently in terms of social media and cell phone utilization. We have video sensing that can get both the affective expression and intra-reciprocity of social interactions. Symptom expression assessment, not in the lab but in the real world, in the context where the person is experiencing their symptoms, as well as ambulatory physiology measures that can be added as well. This has attracted the attention of industry. Industry is creating tools that allow research platforms to accomplish this, such as the Google Verily Health Watch, as well as the Apple Research Toolkit. These have already been deployed in a number of studies, but none of these yet are linked to simultaneous recording of brain activity. Some examples of how these can be deployed in studies. So the way that autism is screened involves a workforce-intensive clinician interview of child behavior, interacting with the clinician, interacting with the parent. Using computer vision approaches, you can assign landmarks to different parts of the face, then anonymize the face by removing the distinguishing features and watch the movement of the facial expression in synchrony with the stimuli that are being presented. This can also be ported into an ambulatory technology on a cell phone, and that's been done with the Apple Toolkit and the app called Autism and Beyond, where the iPhone is presenting the visual stimulus, the social stimulus, the child is sitting on the parent's lap at home, and the camera is watching the child's reaction to the video, again, capturing the landmarks of the face, throwing away the facial identifying information, and doing analyses on the movement and facial expression in response to the stimulus. These technologies can measure how we navigate our world and help to capture symptoms that are only expressed in certain parts of our geography. For example, cues to drug use may occur in certain parts of the neighborhood or when you're interacting with certain individuals. Mobile health technologies allow us to quantify the amount of time that people spend in their neighborhoods and be able to log where and when they experience their cravings or where and when they experience their flashbacks or other episodic symptoms that occur in the real world. Virtual reality paradigms can also bring the real world into the lab so that we can more precisely measure the physiological activity using simulation of real-world experience in the laboratory environment. The correspondence between some of these behavioral quantification measures and standard clinical rating have been studied in studies such as this one, showing significant correlation between, in this case, measures of psychosis and facial expression to a clinical interview. We propose a stepwise plan that will start with a supplementation opportunity to already funded brain initiative studies where we have direct neural recordings of brain activity from circuits that generate behaviors that could be amenable to quantification. The second step would be to convene a meeting sponsored by the NIH to bring together the research communities on behavioral quantification, deep phenotyping, and neural engineering and recording to establish and facilitate team building across these various disciplines. And then the third stage in the following fiscal year would be a funding opportunity announcement to support work in this area. And our ultimate goal is to accelerate the discovery of basic brain science underlying complex human behaviors and ultimately enable intervention development for these leading causes of disability worldwide. And I'm happy to answer questions.
Hi, this is Rafa. Can you hear me?
Yes.
Thank you, Holly. I feel uncomfortable with your proposal, and also I couldn't speak earlier, but I felt also uncomfortable with the proposals about glia or neuromodulation, or I feel that that we're diluting the brain initiative, and as Story was saying earlier, we're losing the focus. So you have to realize that we have what I would call the $6 billion problem, which is that there's $6 billion of yearly neuroscience NIH funding that the Brain Initiative shouldn't be trying to do. And the reason why the Brain Initiative went through is because it had a very sharp focused arrow to the heart of the neural circuit problem by building tools. So I would actually encourage the panel to think about the difference that the Brain Initiative can make in the future, as opposed to just being more incremental research that other parts of NIH can do. And I'm going to say what I always say in every one of these meetings, that at least our vision when we crafted the plan that gave rise to the Brain Initiative is to putting a game changer in the picture and enable an approach to neuroscience which doesn't exist yet, which is similar to what goes on in the physical sciences. Large teams, central laboratories, big projects, bring in the physicists. The nano people are left out of all of this. Bringing the other science that can actually help usher a new revolution, as opposed to just hijack the Brain Initiative and divide it up into small little pots of money for every of the interest groups that are in neuroscience right now. So with all my respect to your plan and to your good will, I think that this may not be the right pot of money to do this, and I would encourage the panel to use this small pot of money compared to the $6 billion to do something transformative.
So I think that Dr. Yusta is making a very important point that bears discussing. The Brain Initiative should be doing things that only the Brain Initiative can do that would radically transform our ability to study and ultimately treat brain health. Our portfolio analysis reveals that in the NIH portfolio, there's only one project that does brain behavior quantification. It is in the Brain Initiative. And I would like to highlight why this is radical. The way that we measure behavior in the rest of our portfolio is generally to use clinical rating scales where we ask the person to subjectively report how they've been spending their time, what they've been thinking about, and how they've been feeling and acting. And we know that, and typically this is integrated over a period of days to weeks. How have you been feeling on average in the past 2 weeks? This is a very low level of resolution, and currently our Brain Initiative projects that do have behavioral outcomes use these very low resolution behavioral measures. What we're seeking to do in this opportunity is to bring those projects up to the state of the art and advance the state of the art so that we can be measuring the behavioral expression of brain phenomenon at the same temporal resolution that we're measuring the brain activity. And the last bullet point on this slide is taken directly from the Brain 2025 report, which is goal number 7, which is to integrate the findings across the prior 6 goals so that we can discover how dynamic patterns of neural activity are transformed into cognition, emotion, perception, and action in health and disease. And I would posit that we cannot achieve that goal unless we measure brain activity and cognition, emotion, perception, and action in health and disease. This will be the only opportunity within the BRAIN initiative and within NIH to accomplish that goal.
Hi, this is Jim Eberwine. I did want to comment. I actually do think that this is an appropriate use of brain monies, because I think we do need to have these quantitative features associated with many modalities of brain analysis as we can get. But I guess my concern is more the unfocused nature of it. Now, essentially any behavior could be studied. This is human. I worry about privacy issues. And one of the other aspects of this is if this were to move forward, I would suggest that there be an additional RFA linked to this for neuroethics evaluation of data and progress that's made in this particular area, because this in particular impacts human behavior. And so I think linking the two would be an appropriate thing to do if this were to move forward.
That's certainly a good idea, and we should and will do that.
I'm wondering if there are other comments in between or in addition to Rafa's on the circuit focus of this proposal. It seems like Jim feels like this is appropriate and Rafa doesn't. I'm wondering if there are other comments in this from other members.
Yeah, so this is Mark Schnitzer. I have to confess that particularly when I saw the suite of these FOAs that we've been talking about, that my thinking ran along the lines of what, Rafa articulated, which is that, we get into thinking about glia, field application of neuromodulation, and now we're studying behavioral monitoring devices. Collectively, there's a very serious and notable, broadening of the scope that may not be evident when you focus on one FOA in particular. Now, sitting here in Silicon Valley, I can say that it's not just Google and Verily which are doing this. There's a very active set of companies and startups which are focusing on these devices. So this is an outstanding research avenue. I think the question for us is, is this kind of work best funded within the BRAIN initiative, or perhaps just with an RFA from NIH more broadly? And I think that's an important question. All these topics that we're looking at that are branching out are very worthwhile. Whether or not they're suitable for the BRAIN initiative, I think, is another question.
Thanks, Mark. Are there others?
It's Brad Hyman. Can you hear me?
Yep.
Okay, good. So I actually think that this has a real possibility of making an enormous impact on relatively short dollars. Translating our understanding and deep understanding of the circuits into any sense at all of how humans behave is critical. And even if the outcome is less traumatic than that, it would be transformative in the context of every kind of social and behavioral psychological study, not to mention, of course, the kind of translational impact that these sorts of technologies would have in terms of running clinical trials in neurocognitive disorders.
Thank you. I appreciate it. I'm going to cut you off just because.
Great.
Yeah, thank you. I think we need to vote on it based on time. Go
Brad,
ahead.
can I make a suggestion? This is
Yeah.
Story. How about if you go ahead with a supplement and the workshop, and then maybe bring it back informed by those two activities? So instead of the whole.
Okay, Story, I think that's a great idea. So we'll hold back then. I don't think do we need approval to offer supplements in this area?
And the workshop.
And the workshop.
And the workshop? Well, if there are any objections to Story's plan, we'll go forward with that then. Thank you.
Okay, thanks very much. And I'm sorry this was obviously we could have had a lot more discussion, but we have to end the open session now so that we could start the closed session. So I would ask you all to hang up or get off of this webinar, and for the federal staff and members of the work group, please dial into the other web information that you also have. Thanks very much.